Welcome to section 3.3, Eukaryotic cells. These types of cells are the building blocks of all complex life, from single-celled protists to massive organisms like whales and sequoia trees. Understanding their internal structures helps us make sense of how life operates on both a microscopic and macroscopic scale. By the end of this lesson, you'll be able to describe the structures that all eukaryotic cells share, distinguish between plant and animal cells, explain the role of the plasma membrane, and summarize the function of each major organelle. We'll also explore the cytoskeleton, the extracellular matrix, and intercellular junctions. Eukaryotic cells are defined by the presence of membrane-bound organelles, including a nucleus. This compartmentalization allows for specialization and efficiency, distinguishing them from the simpler prokaryotic cells. Eukaryotes include animals, plants, fungi, and protists, and their cells are typically much larger and more complex. Plant and animal cells share many organelles, but each also has unique features. Plant cells have a rigid cell wall, chloroplasts for photosynthesis, and a large central vacuole that stores water and helps maintain structural pressure. Animal cells generally lack these features, but include lysosomes for waste breakdown and centrosomes involved in cell division. The plasma membrane is made up of a phospholipid bilayer with embedded proteins. It defines the cell's boundary, maintains the internal environment, and allows communication and exchange with the outside world. This selective permeability is key to homeostasis. Inside the plasma membrane, the cytoplasm fills the space between the nucleus and the membrane. The cytosol, the fluid part of the cytoplasm, acts as a medium where organelles are suspended and cellular processes occur. Think of it as the workspace of the cell. The nucleus houses the cell's DNA and directs its activities. It's protected by a nuclear envelope and includes the nucleolus, where ribosomes begin their formation. It's essentially the cell's command center. Ribosomes are the sites of protein synthesis. They aren't membrane bound and can be found floating in the cytoplasm or attached to the rough endoplasmic reticulum. Both eukaryotic and prokaryotic cells contain ribosomes. The ER comes in two forms. Rough ER is studded with ribosomes and helps process proteins. Smooth ER, on the other hand, synthesizes lipids and helps detoxify harmful chemicals. Together, they form a network of membranes that support cellular manufacturing. The Golgi apparatus modifies, sorts, and packages proteins and lipids for transport. It's like the shipping and receiving center of the cell, sending materials to their final destinations. Lysosomes are full of enzymes that digest cellular waste and worn out structures. Peroxisomes, meanwhile, carry out oxidation reactions, breaking down fatty acids and neutralizing toxic compounds like hydrogen peroxide. Peroxisomes deserve a special note because they form independently and don't bud from the Golgi. Their enzymes help detoxify harmful substances and support metabolic processes by breaking down long chain fatty acids. Mitochondria are the energy producers of the cell. Through cellular respiration, they generate ATP. These organelles have their own DNA and ribosomes, a clue to their evolutionary origins, which we'll explore shortly. Chloroplasts are the site of photosynthesis in plant cells. They contain chlorophyll and internal membranes called thylakoids, where light energy is converted into glucose. Like mitochondria, they have their own DNA and ribosomes. The endosymbiotic theory explains the origin of mitochondria and chloroplasts. These organelles are thought to have originated from free living prokaryotes that were engulfed by ancestral eukaryotic cells. Evidence includes their double membranes, circular DNA, and ability to replicate independently. The central vacuole in plant cells stores water, nutrients, and waste. It also helps maintain turgor pressure, keeping plants upright and structurally sound. The cytoskeleton is a dynamic framework that gives the cell shape, supports organelles, and enables movement. It includes microfilaments for flexibility, intermediate filaments for strength, and microtubules that act like railroad tracks for intracellular transport. Outside animal cells, the extracellular matrix is composed of proteins like collagen and fibronectin. It provides structure, helps anchor cells, and facilitates communication between them. It's critical in tissues where cells must work together. Cells communicate and stay connected through specialized junctions. In animals, tight junctions seal cells together. Desmosomes provide mechanical stability, and gap junctions allow signals and small molecules to pass between cells. In plants, plasmodesmata form channels between cell walls for material exchange. Eukaryotic cells contain a wide variety of structures that work together to support life. Whether it's generating energy, synthesizing proteins, or maintaining structure, every part of the cell contributes to the bigger picture of life's complexity. Understanding these systems helps us see the unity and diversity of life.